Of all your senses, only one skips the checkpoint. Sight, sound, touch, and taste are sorted and relayed before they reach you. Smell goes straight in to the parts of the brain that hold memory and feeling. It is why a scent can put you back inside a room you left 30 years ago before you can name what you're smelling. Tonight we follow that short road. There's nothing to do now. Close your eyes, breathe slowly, and let the world come to you. Somewhere in the last week or so, a smell caught you off guard. Warm dust lifting off a radiator the first evening the heating came on. Soap on the collar of somebody else's coat. The particular sweetness of wet pavement outside a shop doorway. And for a second, before you had any word for what you were smelling, you were somewhere else entirely. A hallway. A back garden. A kitchen at a certain hour of the afternoon, with the light coming in low. That is what we are going to spend the next couple of hours on. Not a lesson exactly, though a little chemistry will drift past now and then. Mostly the quiet strangeness of it. The air is carrying small amounts of the world into you all night long, and your brain has arranged itself so that some of those arrivals go almost directly to the place where you keep your life. There is a reason smell does this, and the other senses mostly do not, and it comes down to wiring. To which cable runs where? When you look at something, the signal from your eye does not go straight to the part of the brain that makes sense of seeing. It stops first at a structure deep in the middle of the head called the thalamus. Think of it as an exchange, a switchboard where things are sorted before they are sent on. Hearing stops there. Touch stops there. Taste stops there. The thalamus filters and weighs and decides what deserves to travel upward, and only then does the message continue to the cortex, where it becomes something you notice. Smell does not take that route. Smell is the only sense that reaches the cortex without being relayed through the thalamus first. The whole journey is short enough to describe in a breath. High up inside your nose there is a small patch of tissue only a few square centimeters across, and in it sit nerve cells that are unusually exposed to the outside air. A molecule drifts in and settles on one of them. The cell fires and passes its signal to the olfactory bulb, a small elongated structure above and behind the top of the nose. That is the first handover. From the bulb, a bundle of fibers runs backward into the brain and hands the signal over a second time, and it lands in 3 places at once: the piriform cortex, which is the first place a smell becomes a smell; the amygdala, which gives experience its emotional color; and the entorhinal cortex, which is the doorway to the hippocampus, where autobiographical memory is assembled. 2 handovers. That is the distance from the air in a room to the machinery of feeling and remembering. None of which means the thalamus is left out of smelling altogether. It gets its turn just later. There is a pathway running onward from the piriform cortex through a part of the thalamus and into the front of the brain, where you do the slower work of judging and naming and deciding whether you like something, So the word arrives eventually. It simply arrives after the feeling has already gone through you. That order, feeling first and label second, is the whole peculiar sensation of being ambushed by a scent. The famous story about all this is not quite the story people tell. In Proust, the moment turns on a small shell-shaped cake dipped in lime blossom tea, and it happens on the tongue rather than in the nose. The sensation he describes is flavor, which is taste and warmth and texture together with the smell that rises inside the mouth. He does not use the word smell at that instant. He comes to it a little later, writing about how smell and taste linger when other things have gone. So the founding anecdote of scent and memory is really about a mouthful of tea. It is a lovelier fact than the tidy version. What researchers have found since is genuinely worth knowing, and it is quieter than the legend. When you cue somebody's memories with a word, or a photograph, or a piece of music, the memories that surface tend to cluster in adolescence and early adulthood, roughly the years between 11 and 25. Cue the same people with an odor, and the pattern shifts. The memories come from further back, from the first decade of life, before the age of about 10. That finding has held up better than almost anything else in this field. Something about the way scent is wired seems to reach past the years you can narrate and into the years you only lived. Those odor-cued memories are also described differently. People rate them as more vivid, more emotional, and accompanied by a stronger sense of having been physically returned somewhere. It is worth being careful here because that is a statement about how the memories feel, not about whether they are correct. Nobody has really tested their accuracy. The field has measured vividness and emotion, which are easier to ask about. So the honest version is this: a smell does not necessarily bring back a truer picture of your grandmother's kitchen. It brings back one that arrives with the force of being there. You do not need to hold on to any of it. The wiring works whether or not you know about it, and it has been working every time a smell has caught you off guard. For now it is enough to lie still and let the idea sit, that of all the ways the world gets into you, this one took the short road. There is a number that has followed your nose around for about 100 years, and you have almost certainly met it. 10,000 smells. It turns up in textbooks and on the little printed cards beside perfume counters, always with the same quiet confidence, as though someone once sat a great many people in a room and counted. Nobody did. The figure comes from two Americans, Ernest Crocker and Lloyd Henderson, working in 1927 on the practical problem of how industry might describe an odor. They proposed that any smell could be scored on 4 qualities: fragrant, acid, burnt, and a fourth they called caprylic, the goaty, fatty note of old cheese and warm skin. Each quality was rated from 0 to 8, which gave every smell a 4-digit code, and there were just over 6,500 codes to go round. Crocker later rounded that up to 10,000, because 10,000 is a friendlier number, and the rounded version escaped into the world and lived there for a century. It was never a measurement. It was the size of a filing system. The strange thing is how small it sounds once you sit with it. 10,000 is roughly the number of words in comfortable everyday use. It seems far too modest for something that can tell one apple from another, or notice that the coat by the door has been out in the cold. So in 2014, a group at Rockefeller University tried to actually measure it. They took a library of 128 odorous molecules and blended them into mixtures of 10, 20, or 30 components so that a single smell became a chord rather than a note. Then they asked a couple of dozen volunteers to say which of 3 vials was the odd one out. Most people could tell 2 mixtures apart when they shared less than about half their ingredients, and from that small result the researchers worked outward to the whole library and arrived at a headline: humans can discriminate at least 1 trillion olfactory stimuli. It was a wonderful number, and it traveled fast. Within a year, 2 separate papers took the calculation apart, politely and thoroughly. One showed that the same data, with slightly different but equally reasonable choices about how to handle it, produced answers ranging from a few thousand at the low end to a number 30 digits long at the high end. It also pointed out that the formula, read correctly, gives an upper limit rather than a lower one, so the honest reading of the experiment is at most a trillion, not at least. The other paper did something simpler. It applied the identical method to color vision, where the answer is already known from a century of careful work. The method predicted that we can distinguish more than 1 billion colors. The measured figure is somewhere between 1 and 2 million. The original authors replied, and the title they chose for their reply was that humans can discriminate trillions of olfactory stimuli, or more or fewer. There is a kind of grace in that. It concedes in 4 words that the ceiling has not been found. And that is where the matter still rests. Only 148 pairs of smells were ever actually tested. The true number is not known, and it may not be the kind of thing a number can hold. Smells do not sit in tidy rows waiting to be counted. What the experiment genuinely established is that the old figure was far too small. Whatever you are capable of, it is vastly more than 10,000. Underneath both numbers sits an older assumption, and this one is worth putting down gently. For most of the last 2 centuries, educated opinion held that humans are simply bad at smelling, that we traded the nose for the eye somewhere back along the line, and that the sense we kept is a vestige. That belief was traced in a review published in 2017 to a single 19th century anatomist, Paul Broca. Broca was not measuring anyone's sense of smell. He was arguing that human free will required a brain less enslaved to odor, and he inferred a shrunken olfactory bulb to match the theory. The idea passed into Freud's writing, and from there into general knowledge, and went unchallenged for something like 150 years. The anatomy does not support it. The human olfactory bulb is only 5 or 6 millimeters across, but it is large in absolute terms and holds a number of neurons broadly comparable to other mammals. Across roughly 24 mammal species whose body weights span nearly 6,000-fold, the neuron count in the bulb varies only about 28-fold, and we sit comfortably in the middle of that range. Our bulbs are smaller than a dog's and larger than those of mice and rats, which nobody has ever accused of smelling poorly. And where thresholds have been measured properly in both species, the picture is not a ranking but a division of labor. Of the 15 odor molecules for which humans and dogs have both been carefully tested, humans are the more sensitive to 5 of them, and those 5 are components of the smells of fruit and flowers. Against rats, humans win on 31 of 41. Ripe peach, warm blossom, the skin of a lemon under your thumb. Whatever we lost, we did not lose that. High in your nose, above the wide, easy channel you breathe through without thinking about it, there is a small patch of tissue on either side. Nothing about it looks important. And yet the instructions for building it take up more room in your genetic material than almost anything else you carry. The proteins that do the sensing are called odorant receptors. Each one is a folded shape threaded through the wall of a nerve cell, arranged so that some molecules settle into it and others drift past. Your genome holds somewhere around 870 places where such a receptor is written down. Of those, roughly 400 still work. That is the largest single family of related genes in the mammalian genome, a few % of all the genes you have, which is a generous share of the instruction manual to spend on the air. The rest of the family is quiet. Geneticists call them pseudogenes. The text is still there, but a letter has changed somewhere or a fragment has slipped out of place, and no working protein can be built from what remains. They are genetic fossils, the outlines of receptors your ancestors once used, pressed into the genome the way a shell is pressed into stone. Mice keep over 1,000 of theirs in working order. We have kept fewer, and we have kept them a very long time. None of this was known until 1991. Linda Buck and Richard Axel had spent about 8 years hunting for these receptors, reasoning their way towards what such a protein would look like before anyone had seen one, working with tissue from the noses of rats. What they published was not the whole family. It was 18 members of it, enough to show that the family existed and that it was enormous. They had found the door, not the house behind it. In 2004, they were given the Nobel Prize in Physiology or Medicine for their discoveries of odorant receptors and the organization of the olfactory system. There is a rule inside that patch of tissue that seems slightly improbable when you say it out loud. Each individual sensory neuron up there settles on one receptor gene and expresses only that one. Millions of cells, each listening for a single kind of shape, all pressed together in the same thin sheet. 400 kinds of listener does not sound like enough for a world. In 1999, Bettina Malnick, working with Buck and others, showed how the arithmetic manages it. They found 3 things at once. One receptor responds to a number of different molecules. One molecule is picked up by a number of different receptors. And different molecules switch on different combinations of them. A smell is not a note struck on a single key. It is a chord. 400 receptors are no more 400 smells than 26 letters are 26 words. What reaches the brain is a pattern, a particular set of receptors lit together at particular strengths, and the shape of the whole pattern is what means wood smoke, or wet wool, or a peeled orange, which leads somewhere quietly strange. Those receptor genes vary from person to person, and they vary far more than most of our genes do. In 2014, Joel Mainland and colleagues went through the human repertoire looking for differences that actually change how a receptor behaves. Around 63% of the ones they examined carried such variations. No two people, as far as anyone can tell, carry quite the same working set. So the person lying beside you or in the next room is not smelling quite what you are smelling. Not a different intensity. A different chord played on a slightly different instrument from a family of genes no two of us have inherited in the same arrangement. There is nothing to resolve in that. It is simply how it is, and it has been true in every room you have ever fallen asleep in. Quietly, at some point in the last hour, and without consulting you, your body chose a nostril. One side of your nose is doing most of the breathing right now. The other is partly closed, taking a smaller share of the air, resting. You have almost certainly never noticed, because the total feels the same and the arithmetic works out. But if you were awake enough to test it, and there is no need to be, you would find that the two sides of your nose are rarely equal partners. Most of the time one leads and one follows, and after a few hours they quietly swap. This is called the nasal cycle, and it was described in the 1890s by a German physician named Richard Kaiser, who noticed that the swelling inside the nose kept moving from side to side in perfectly healthy people. It is not congestion in the sense of a cold. It is a rhythm, and it is running in you tonight. The machinery behind it sits just inside each nostril. The inside of the nose is not a smooth tube. It is folded into shelves of thin bone called the turbinates, curled like scrolls, wrapped in soft tissue that is unusually rich in blood vessels. That tissue is erectile. Not in any dramatic sense, simply in the plumbing sense. It can fill with blood and swell, or drain and shrink, and when it swells it narrows the passage beside it. The autonomic nervous system holds the tap. Sympathetic signals tighten the vessels and open the airway. When they ease, the tissue engorges and the airway narrows. The two sides are held in a slow seesaw, one opening as the other closes, so the overall resistance of the nose stays roughly steady while the balance between the halves keeps sliding. How long a swing lasts varies a great deal. Sources give anything from under an hour to several hours, and plenty of people show no tidy rhythm at all or a pattern that wanders and reverses. It shifts with posture too. Lying on one side tends to congest the nostril nearer the mattress and open the upper one, which is part of why turning over in the night can make your breathing feel suddenly pleasantly clearer. None of which would matter much for smell, except that airspeed changes what you can smell. odor molecules do not simply float past the sensing surface. They dissolve into the thin layer of mucus and are absorbed by the tissue, and some do this eagerly while others are reluctant. A molecule that sticks readily will be captured near the front of the airstream. A molecule that resists will travel further before it is taken up. Move the air quickly, and the sticky ones are carried deep enough to spread across the whole sensing surface instead of being caught at the front. Move it slowly, and the reluctant ones finally have time to be absorbed and read. Fast air and slow air are, in effect, two slightly different instruments, which means your open nostril and your quiet nostril are not sampling the same world. In the late 1990s, Noam Sobel and colleagues published exactly this result under a title worth remembering: The World Smells Different to Each Nostril. Present the same mixture to both sides and people can report a subtly different impression from each, and which side gives the richer reading depends on the mixture. Because the nasal cycle keeps turning, the pairing keeps changing, and over the course of a day each of your nostrils gets to be both instruments. You are not aware of any of this. What arrives in consciousness is one smell, single and whole, the way you hear one sound rather than two ear reports. The comparison is done somewhere below the level you can inspect. The two nostrils also sit a little apart, sampling air from patches of the world roughly 3.5 centimeters from each other. That is a small baseline, and smell is a slow, drifting signal compared with sound, so directional smelling in humans is crude at best. With a pure odor, one that does not tingle or bite, most people cannot reliably say which nostril received it. Still, the two streams are worth something. When researchers at Berkeley blindfolded volunteers, plugged their ears, and set them to follow a chocolate-scented string laid through the grass on hands and knees, people could do it, and they got better with practice, and they did it faster and with fewer wanderings using two nostrils than one. The comparison is faint, but it is there. And the sniff itself is part of the sense. Air does not enter the smelling region on its own. Without a sniff, there is essentially nothing to detect, which is why you lean in and draw breath before deciding whether the milk has turned. A sniff is a deliberate act of sampling, and it is remarkably quick to adjust. Within a fraction of a second of the odor arriving, before you could name what you were smelling, the shape of the sniff has already changed, shorter and shallower for something strong or unpleasant, longer and fuller for something faint or agreeable. The nose withdraws from a smell almost before you know you dislike it, and a single natural sniff seems to gather most of what is available. Repeated sniffing feels thorough, but it adds surprisingly little. So tonight, without effort, you have sampled the air of this room a few hundred times. One nostril did the greater share of it, and read the world at speed, while the other held back and read it slowly, and at some point, unannounced, they will change places. The word for it is younger than you might expect. Until 1964, there was no good name in English for the smell of rain arriving on dry ground. Chemists called it the argillaceous odor, meaning roughly the smell of clay, and nobody has ever said that out loud with any pleasure. Then two Australian chemists, Isabel Joybear and Richard Thomas, published a short paper in Nature and gave it the name we use now, Petrichor. They built it from two Greek words, petra, meaning stone, and ichor, the thin bright fluid said to run in the veins of the gods in place of blood, an essence drawn out of rock. It is a rather grand name for something that costs nothing and arrives unannounced, and it has lasted because nothing better has come along. What they did was more patient than the name suggests. They gathered rocks and soils that had been sitting through warm, dry weather, and they steam distilled them, and out of the stone came a small quantity of yellowish oil. That oil is the smell. It is not one compound, but a mixture, mostly fatty acids and alcohols and hydrocarbons that plants quietly shed into their surroundings. During a dry spell, those molecules settle onto mineral surfaces and stay there, held most readily by clays and by materials rich in silica. Nothing washes them away, so they accumulate. A long, dry summer is, in a sense, the ground slowly being loaded with scent, week by week, waiting. There is a strange footnote to this. The following year, the same two chemists published a second paper showing that these same compounds slow the germination of seeds and hold back the earliest growth of seedlings. The oil that smells so hopeful to us is, to a seed, a break. Dry ground keeps its seeds waiting, and the rain that carries the smell away is also the rain that releases them. Long before any of this was written down, perfumers in Kanauj in northern India were baking discs of clay and distilling the scent off them into sandalwood oil. They called it Matika Attar, the perfume of the earth. They had the smell in a bottle centuries before anyone had it in a paper. How the scent gets from the ground into the air took longer to work out, and the answer came from a high-speed camera. In 2015, two engineers at MIT, Young-Soo Zhang and Cullen Bui, ran something like 600 filmed experiments across 28 different surfaces, soils among them. Watch a single raindrop land on porous ground, slowed right down, and you see something like a small fizzing. As the drop spreads, it pins tiny pockets of air against the surface. Those bubbles rise up through the body of the drop and burst at the top, and each burst flings out a fine spray of droplets, hundreds of them, in a few millionths of a second. The team colored their water with dye and found the dye riding up in the mist, which tells you these droplets can carry whatever the ground had dissolved in it. The part of that work worth keeping is quietly counterintuitive. The heaviest rain does not make the most of this mist. Light and moderate rain make far more of it. A downpour makes much less. The gentle rain, the sort that begins as a few slow taps on the path, is the fragrant one, which is something you may already know without knowing it. The smell is strongest in the first few minutes, before the weather really commits. Mixed into all of this is a second molecule from a separate discovery by different people a year later. In 1965, Nancy Gerber and Hubert Le Chevalier isolated an earthy-smelling oil from soil bacteria and named it geosmin, from the Greek for earth and smell. It comes largely from Streptomyces filamentous bacteria living in almost every handful of soil on the planet. A single gene makes it, and as far as anyone has tested, every Streptomyces species carries that gene. They differ wildly in almost everything else they produce. They agree on this one smell. We are extraordinarily good at noticing it. Reported thresholds vary a great deal between studies and between people, from a few parts per trillion in water up to 100 or so, which still puts geosmin among the small handful of things our noses detect at almost absurdly low concentrations. There is a further refinement, easy to love. Geosmin comes in two mirror image forms, and the one that bacteria actually make is the one we are tuned to. In one careful study, our threshold for the natural version was around 11 times lower than for its mirror twin. We are not merely sensitive to Earth. We are sensitive to the exact-handedness of it. For a long time, the smell was assumed to be an accident, a waste product we happened to notice. The better evidence says it is a message, though not one addressed to us. In 2020, researchers found that the genes for geosmin fire only when the bacteria are making spores under the same controls as sporulation itself. Springtails, tiny soil animals, respond to geosmin in their antennae and walk towards it. They graze on the colonies and carry the spores away, stuck to their bodies and passed in their droppings. The bacterium is advertising, and the springtail is the courier. Whether our own sensitivity means anything is an open question, and the honest answer is that nobody knows. You have almost certainly tasted geosmin too. It is in beetroot, which is why beetroot tastes of soil to some people and of sweetness to others. A lawnmower starts somewhere down the street, and before you have thought about it at all, you know roughly what month it is. That green, sharp, faintly sweet smell arrives ahead of every other detail. It is one of the fastest chemical messages in the living world, and it is genuinely a message, though not quite the one people usually describe. When a leaf is torn, the damage begins in the membranes of its cells. Plant membranes are built partly from long fatty acids, and one of them, linolenic acid, is the raw material for this smell. An enzyme called lipoxygenase adds oxygen to that fatty acid, and a second enzyme, hydroperoxide lyase, cuts the resulting molecule in two. What falls out of the break is a small 6-carbonaldehyde, hexanol, light enough to leave the leaf immediately. Some of it is converted into the matching alcohol, hexanol, and some of that is dressed up further as an ester, hexenyl acetate. Those three, in slightly different proportions, are the smell of cut grass. Chemists call the whole family green leaf volatiles, which is one of the rare scientific names that describes exactly what it smells like. The speed is the quietly remarkable part of it. The enzymes are already sitting inside the leaf, kept apart from their raw material by the membranes themselves, and the wound is what introduces them to each other. There is no manufacturing delay, nothing to wait for. Within seconds of a blade passing, the air above the lawn has changed. Almost every land plant can do this. A path trodden through a meadow, a hedge clipped in the afternoon, a bruised nettle, a bag of salad leaves opened over the sink, all of it is the same reaction, running at slightly different settings. It is worth being careful about what the plant is actually doing, because this story often gets told with more drama than the evidence supports. Green leaf volatiles are not an alarm aimed at caterpillars in particular. Any wound releases them: hail, a footstep, a mower, a grazing deer, your own thumbnail. What the plant does with them afterwards is less settled than it sounds. Some of these compounds seem to slow the growth of bacteria and fungi at the raw edge of a wound, which would make a certain kind of sense, since a cut leaf is an open door. And there is good laboratory evidence that plants downwind of a damaged neighbor become quicker to mount their own defenses afterwards, as though the smell had put them on notice. Whether that makes a measurable difference out in a real field across a real season is still argued over. Researchers who have gone looking for the payoff in the wild, in seeds set or offspring raised, have found it thinner than the laboratory work would suggest. The clearest piece of the puzzle came from maize. In 1990, a group of researchers found that corn seedlings being eaten by caterpillars release a second class of volatile, the terpenoids, heavier and more resinous than the green leaf compounds, and that these carry far enough to be found by small parasitic wasps, which lay their eggs in the caterpillars. The detail that makes it convincing is what happens when you damage the seedling yourself. Cut it with a blade and the terpenoids do not come. Apply the caterpillar's own oral secretions to the same cut, and they do. The plant is not simply leaking when injured, it is answering something specific in the mouth of the thing eating it. The wasps, for their part, learn. They are not born knowing what a chewed corn plant smells like. They associate the scent with a successful search and go looking for it again. That word, terpenoid, is the bridge to a great deal of the outdoor world you already know. The terpenes are the resin family, the smell of a pine wood in the sun, the sharp clearing note of eucalyptus, the oily green scent left on your fingers after brushing past a tomato plant. Tomato leaves are worth pausing on. The smell does not drift off the leaf surface on its own. It is held in tiny brittle hairs along the stems and the undersides of the leaves, and it takes a small collision to break them open. You cannot really smell a tomato plant from a distance. You have to touch it, and then your hand keeps the smell for an hour. Terpenes are also much of the reason a wood smells so much bigger after warm rain. These molecules are volatile, but only moderately so. They sit in needles and resin and leaf oils, and how readily they leave depends a great deal on temperature. Warm air lifts more of them into the open. Rain wets every surface in the forest at once, and then the sun returns and the whole canopy begins to dry, and for an hour or two afterwards, the air carries a concentration of pine and leaf oil that a still dry day would take far longer to build. Damp air seems to hold scent better than dry air as well. Standing in the middle of that is not a subtle experience. The trees are, in the most literal sense, evaporating. None of it was arranged for you. The grass is not addressing you. The pine is not perfuming the hillside on your behalf, and the tomato plant would rather you left it alone. What reaches you is a private conversation between organisms that have been talking chemically for a very long time, and which you happen to be equipped by some accident of your own biology to overhear. Somewhere tonight, at some table, a person is quietly moving a scatter of green leaves to the rim of their plate. To them, those leaves taste of soap. Not faintly, not as a turn of phrase, but plainly and unmistakably the way soap tastes if you ever got a little of it in your mouth as a child. Across the table, someone else is eating the same dish and finding the same leaves bright and citrusy and green, and wondering, gently, what all the fuss is about. Neither of them is mistaken. They are simply not receiving the same message. Coriander leaf, which North America calls cilantro, releases a group of molecules called aldehydes. Several of them are medium-chain aldehydes, decanol, dodecanol, and one called E2-decenol. Aldehydes of this same broad family have long been part of the perfumer's cupboard, including the fragrances built into soaps and detergents, which is why the comparison people reach for is not fanciful at all. Some noses really are picking out the note that soap and coriander have in common, and reporting it as the whole experience rather than one thread in it. For years, that was as far as the story went. Then, in 2012, a consumer genetics company put a simple question to a very large number of people. Does coriander taste soapy to you? Nearly 15,000 answered in the first round, and almost 12,000 more in a second group used to check the result. When the answers were laid against the genome, one small region stood out on chromosome 11, sitting close to a cluster of olfactory receptor genes. The nearest of them is called OR6A2, and it has carried the nickname ever since. The Honest Version Parts Company here with the version that circulates. That genetic marker explains only about half of 1% of the difference between people. Take every common genetic variant the study could measure, added together, and you still account for something like 9%. Which means that more than 90% of why you feel the way you feel about coriander lies somewhere else entirely, in what you were fed as a child, in what your family cooked, in whether the leaf arrived as an ordinary part of dinner or as an ambush in an unfamiliar dish. The gene is real. It is just a whisper, not a verdict. The receptor itself has since been caught in the act. Recent work has shown that human OR6A2 does indeed respond to precisely those medium-chain aldehydes, latching onto them with a reversible chemical handshake. So the biology holds up beautifully at the level of a single protein, while remaining almost negligible at the level of a whole person. Both things are true at once, and it is worth sitting quietly with that. The numbers behind the phrase you may have heard are worth softening too. In that same survey, the proportion who described coriander as soapy ran from under 4% among people of South Asian descent to about 14% among people of Ashkenazi descent, with most other groups landing somewhere between 8 and 13. Not 1 in 5, then, and not the same anywhere. The lowest figure belongs to the cuisine where coriander is most ordinary, which tells you something about how much familiarity does. Coriander is only the most famous of these small private differences. There is a molecule called androstenone, a steroid that turns up in sweat and in pork. Ask a room of people what it smells like, and you will get 3 answers, not 1. Some find it sharply unpleasant, sweaty or urinous. Some find it sweet and faintly floral, close to vanilla. And some find nothing there at all, an odorless puff of air. That 3-way split traces in part to variants in a receptor gene called OR7D4. 2 small changes in the protein blunt the receptor, and people carrying the blunted version tend to find the smell weaker and less objectionable. How many people cannot smell it depends entirely on how strong you make the sample and how you ask, and published estimates run all the way from a few percent to most of the room. Roughly 1/4 to 1/3 is a fair, cautious figure. Then there is asparagus. A study published in a medical journal's Christmas issue, drawing on nearly 7,000 people, found that 58% of the men and roughly 62% of the women said they simply could not smell anything unusual in their own urine afterwards. The associated genetic signals sat once again in and around a cluster of olfactory receptor genes, this time on chromosome 1. The important detail is that everyone makes the compounds. This is not a difference in bodies. It is a difference in noses. Violets belong in the same company. The scent we call violet comes largely from a compound called beta-ionone, and the evidence suggests people vary in how strongly they register it, some finding it lush and unmistakable, others catching almost nothing. Which may be why the flower has such a reputation for shyness. Put all of this together, and the picture is a quiet one. When researchers compared people's odorant receptors gene by gene, they found that any 2 of us differ in a way that actually changes how the receptor works at more than 30% of them. Not 30% of our genes, 30% of the ones for smelling. You and the person beside you are standing in the same kitchen, breathing the same air, and living in slightly different worlds. There is a small experiment that works with anything sweet in a bowl by the sofa. Pinch your nose closed, put a jelly bean on your tongue, and chew. What arrives is sweetness, and perhaps a little sourness, and nothing else at all. No cherry. No lemon. No buttered popcorn. Then let your fingers go, and the flavor floods in all at once, as though a light had been switched on in a dark room. The sweetness was never the flavor. The sweetness was only the floor the flavor was standing on. Your tongue is a fairly blunt instrument. It reports sweet, salty, sour, bitter, and savory, umami, the taste of broth and ripe tomato and long-aged cheese, and researchers still argue politely about whether fat deserves a place on that list. 5 words, perhaps 6. That is close to the whole vocabulary of the mouth. Everything else you have ever called taste, The rosemary, the orange peel, the coffee, the particular character of the stew someone made for you once is smell arriving from the other direction. Because your nose has two doors. The one you know about faces the world. You lean over a cup and draw air in through your nostrils, up to the patch of sensing tissue high in the nasal cavity, and you smell the coffee before you drink it. The second door is behind your soft palate, where the back of the mouth opens into the back of the nose. Warm food in a warm mouth is constantly shedding molecules into the air trapped there, and every time you swallow and every time you breathe out, that air is pushed upward past the same sensing tissue. The molecules arrive at the same receptors. Only the route is different. The first way is called orthonasal, the second retronasal, and the second one is doing most of the work at dinner. What makes it strange is that you have no sense of it happening. The brain files retronasal smell under the mouth. It takes a signal from high in your head and hands it to your tongue, and you accept the delivery without question and say the soup tastes of thyme. There is evidence that the brain does not treat the two roots as quite the same thing. That a smell can have one character sniffed from the outside and another from within, which is why some cheeses are far easier to eat than to lean over. But the illusion of location is total. Nobody has ever tasted anything in their nose. Chewing works as a kind of bellows. Teeth break cell walls and release oils that were locked in place. The heat of the mouth lifts them into vapor. The tongue moves, the jaw moves, the throat opens and closes, and the aroma is pumped up the back of the passage in small pulses. Those pulses are timed to your breathing. You do not smell your food continuously. You smell it in waves, mostly on the out-breath, and just after swallowing, which is why the flavor of something seems to bloom a moment after you thought you had finished with it. Slow chewing makes more of those waves. This is not advice, only an observation about plumbing. It also explains the flatness of eating with a cold. Nothing has happened to your tongue. Sugar is still sweet and salt is still salt, and if you concentrate, you can find them. What has gone is the airflow. The passages upstairs are swollen, and the vapor cannot reach the sensing tissue, so the second door is shut, and the meal is reduced to its 5 plain words. People almost always describe this as having lost their sense of taste. They have not. They have lost the far larger part of it that was never taste at all. And then, quietly, a third thing joins in, which is neither taste nor smell. The 5th cranial nerve, the trigeminal, carries touch, temperature, and irritation from your face, and it has endings throughout your nose and mouth. Menthol does not make anything colder. It opens the same channels that cold air opens, and the mouth reports a chill that no thermometer would find. Chili does the reverse, tripping the machinery of heat so that a pepper genuinely burns without burning anything. The prickle of a fizzy drink is a chemical sting rather than a bubble bursting. The rasp of mustard, the dry pull of strong tea against the inside of your cheek, the sharpness at the back of the nose when you slice a raw onion, None of these are flavors in any strict sense. They are your face reporting an event. So a mouthful of soup on a cold evening is at least 4 things happening in convincing unison. Taste from the tongue. Smell from behind. Touch and temperature and the faint chemical bite. Texture too, and even sound, the crunch of a crust reaches you partly through the bones of your skull. All of it converges somewhere behind your eyes and comes back to you as a single thing with a single name. Buttered toast. Tomato. The tea you drink at this hour. It is worth sitting with at the edge of sleep. The great warm, remembered flavors of your life were never really in your mouth. They were assembled from the inside by a nose you were not consciously using, out of air you were about to breathe out anyway. Ask a room full of people what comfort smells like, and the answers come back remarkably similar. Wood smoke. Bread in the oven. An old book opened under a lamp. Beeswax on a wooden table. Cedar in a drawer. Vanilla. On the surface, these have nothing to do with one another. One is fire, one is baking, one is paper that has been quietly aging since before anyone in the room was born. Underneath, though, several of them are doing the same slow chemistry, and the shape of it is worth knowing. Start with the smoke. Wood is held upright by a substance called lignin. It is the stiffening in the cell walls, the reason a tree can stand against wind for 200 years instead of flopping over like a wet stem. Lignin is not a neat repeating molecule, but a tangled, irregular network, and when heat pulls it apart, it does not break into one thing. It breaks into hundreds of small fragments, and a good many of those are light enough to leave the fire and drift across a garden on a cold evening. The one that matters most is called guaiacol. Chemists who catalog smells for a living describe its odor in 2 words, clove and vanilla. That is the standard descriptor for the molecule, not an embellishment. The smell you have been calling wood smoke all your life has, at its center, something that a flavor laboratory would file next to a spice rack. There is a second family from lignin built on a molecule called syringol, and it is the reason different woods do not smell the same. The lignin of soft woods like pine is built mostly from one kind of unit, and it yields the guaiacol side of the family. Hardwoods like oak and beech carry a high proportion of a second kind of unit, and those give the syringols. So a pine fire and an oak fire are not the same smell wearing different clothes. They are chemically different smokes. You might assume syringol does the heavy lifting in hardwood smoke, since there is so much of it. It does not. Odor thresholds tell you how little of a molecule your nose needs before it notices, and by that measure, guaiacol is roughly 150 times more potent than syringol gram for gram. Syringol fills in the background. Guaiacol carries the character. Around them sit smaller notes that analysts have picked out of condensed hardwood smoke and named in plain kitchen language. Something buttery, something toasted, something faintly ashy, something a little cheesy. Read the list and you're reading a description of a good meal cooked outdoors. Now the bread. When a loaf goes into the oven, the inside stays wet and never gets much above the boiling point of water. The crust is different. It dries, it gets hot, and in that heat the amino acids and the simple sugars begin to react with one another. That reaction is named after the French chemist who described it, and it is the reason brown things smell better than pale things. Out of it comes a molecule with an unlovely name and an extraordinary presence, 2-acetyl-1-pyrroline. Two German chemists identified it in the crust of wheat and rye bread in the mid-1980s. Your nose can pick it out of the air at concentrations of a few hundredths of a nanogram per liter, which is to say almost nothing at all. That same molecule, the same one, not a cousin of it, is what makes jasmine rice smell like jasmine rice. It is in basmati. It is in popcorn. It is in pandan leaves, which is why a Southeast Asian dessert can smell obscurely of fresh bread to someone who grew up in a European kitchen. In fragrant rice, it accumulates because of a change in a single gene quietly in the field without any heat at all. In your crust, it is manufactured in the oven in the last half hour. Two completely different roots, arriving at exactly the same smell. And then the books. A study in London analyzed the air above dozens of historical papers from the 19th and 20th centuries. The researchers' own summary of that smell was grassy notes, with a tang of acids and a hint of vanilla over an underlying mustiness. The vanilla is real, and it is lignin again. Cheap wood pulp paper is full of the stuff, and as it slowly comes apart, it releases acetic acid and hexanol and furfural and vanillin, which is not merely vanilla-like, but is chemically the very same molecule that gives a vanilla pod its smell. Some of what you're breathing in a second-hand bookshop is almond, from benzaldehyde, and something faintly floral from the resin once used to size the paper. So the book smells of vanilla because it is coming undone. The smoke smells of clove because the tree is coming undone rather faster. The crust smells of the crust because sugar and protein have rearranged themselves in the heat. Beeswax and cedar have been taken apart too. Cedar's warmth is largely cedrol and the cedrines, but a warm beeswax candle and a cedar-lined drawer are still mostly described the way you would describe them, in honey and pencil shavings and dry sweetness. But the pattern holds well enough. The smells we call cozy are, very often, the smell of something solid being gently and slowly taken apart, usually with a little warmth involved. Wood into smoke, flour into crust, a book into its own long, patient ending. It is a strange thing to find at the center of comfort, and a rather beautiful one. In almost any chemist's shop, there is a shelf of small dark bottles, and somewhere on that shelf there is lavender. It is the one people reach for without needing a reason. Lavender in a pillow. Lavender in a drawer of folded sheets. Lavender in the water at the end of a long day. The plant itself is modest, gray-green and dry-leaved, happier on a stony hillside than in good soil, and yet it has been carrying the job of calm for a very long time. Which makes it worth asking, gently and without needing a tidy answer, what has actually been measured. The molecule usually named is linalool, a light, sweet, woody alcohol that lavender shares with bay leaves and rosewood and many other plants. In rodents, linalool does appear to do something. The animals move less. They settle sooner. The mechanism people propose involves GABA signaling, which is the brain's main way of quietening itself down. That work is real, and it belongs where it was done, in animals and in dishes. Whether breathing a little lavender in an ordinary room produces anything comparable in a person has not been shown. A mechanism is a story about how something might work. It is not evidence that it does. There is a stranger fact underneath all this. The best controlled human trials are not about smelling it at all. They test an oral preparation, a lavender oil capsule swallowed whole. The largest of them enrolled over 500 people and reported a modest benefit against placebo. And here is the detail that tells you what kind of study it was. The placebo capsules were themselves scented with about 1,000th of the lavender oil so that nobody could work out which group they were in by the smell of the bottle. The researchers went to some trouble to remove the fragrance from the experiment. Whatever that trial found, it was not a finding about aromatherapy. The evidence base around that capsule is also narrower than its reputation. Only about 6 central trials exist, most of them from the same small group of researchers, and all of them tied in one way or another to the company that makes the capsule. None of this is scandal. It is simply what a thin literature looks like, and thin literatures tend to shrink when other people repeat them. The inhaled research has a deeper problem, one no amount of funding could fix. You cannot blind a smell. In a drug trial, you can make 2 identical pills. You cannot make a lavender that does not smell of lavender, so everyone in the room, participant and researcher alike, knows exactly which condition they are in, and everyone arrives already knowing what lavender is supposed to do. Expectation gets to sit in the results, and there is no clean way to lift it out. When large reviews have tried to pool what exists, the picture stays faint, a scatter of small studies, results that point in different directions, and evidence the reviewers themselves grade as very low. That is not a verdict that fragrance does nothing. It is a verdict that we have not yet done the work well enough to know. That is not a verdict that fragrance does nothing. It is a verdict that we have not yet done the work well enough to know. Vanilla has been through the same ringer, and the results are worth being precise about because vanilla is the scent most often credited with slowing a heart. In one study, nearly 60 people waiting for a hospital scan were given either a vanilla-like fragrance or plain humidified air. Those who had the fragrance reported feeling markedly calmer, something like 2/3 calmer. Their pulse and heart rate showed no statistically significant difference. In separate work on the startle reflex, a whiff of vanillin tended to soften the involuntary blink people make at a sudden loud sound, but heart rate and skin conductance were again unmoved. A pattern runs through all of it. What shifts is what people say. What holds steady is what the instruments read. The idea of a scent that slows a heart rate seems so far to describe a feeling rather than a measurement. And a feeling is not nothing. It may be most of the story. A smell does not arrive neutral. It arrives carrying whatever was in the room the last 100 times you met it, and for a great many people lavender has only ever appeared at the soft end of the day, in warm water, in clean linen, in the last hour before the lamp goes off. Vanilla has been in kitchens and in the hands of people who were looking after you. The scent is not acting on the nervous system so much as reporting on where you are and what tends to happen next. The ritual may be doing the rest, stopping, running the bath, opening the drawer, choosing deliberately to spend a few minutes doing nothing useful. Those minutes would probably work without the bottle, and the bottle is unlikely to work without the minutes, but they have been arriving together for years now, and the smell has become the way the evening announces itself. So the honest version is quieter than the label on the bottle. Nobody has demonstrated that lavender slows you down. What is demonstrated over and over in your own life is that you have taught yourself what it means. There is a smell in your home tonight that you cannot detect. It is not faint and it is not hidden. It is the house itself. The particular blend of wood and paint and fabric, of soap and cooking and warm dust and the people who live there. And every visitor meets it in the first second through the door, forms a quiet impression, and loses it too. You alone are excluded from your own house. You have lived inside that smell so long that your brain has stopped mentioning it to you. This is adaptation, and it happens far faster than you might expect. Hold a cut lemon under your nose, and the brightness of it fades surprisingly fast, even though the lemon has not changed and the molecules are still arriving. The odor is still there. The report of it has been turned down. For a long time, the assumption was that the nose simply tires, and there is something real at that level. The receptors sitting in the fine hairs of your olfactory cells begin damping their own internal signaling within about 1/10 of a second of being stimulated, a chemical shrug almost immediate. But when researchers went looking for the fading in humans directly, by recording the electrical response from the smelling surface high in the nose, while people rated what they were experiencing, the two measures came apart. Across repeated short exposures, the ratings fell steadily. The signal at the epithelium did not significantly fall at all. Only a minority of volunteers showed any reduction. The nose was still reporting faithfully. Somewhere further in, the message was being set aside. So it is better to say the brain stops passing the smell on than to say the nose gets tired. And once you put it that way, the logic of it becomes rather beautiful. A sense organ that tells you what is constantly present is telling you almost nothing. What matters is what has changed, the new thing in the room, the thing that was not here a moment ago. Steadiness is not news. Your olfactory system spends its energy on edges and arrivals and lets the unchanging background dissolve into the wallpaper. It is not a failure of the sense, it is the point of it, which is why the smell of your home is returned to you briefly when you have been away. Come back after a fortnight and you get it back for a moment. You put down the bags in the hall, and there it is, unmistakable and slightly strange, a smell you have never consciously known and yet recognize completely. Then it dims, and by the time the kettle is on it has gone again, and no effort of attention will bring it back. The specificity of this has been measured. In one study, volunteers lived for 2 weeks with a particular odorant present continuously in their homes. Afterwards, their sensitivity to that odor had genuinely dropped. It took more of it before they could detect it at all, and it seemed weaker when they did. But their sense of smell in general was untouched. Everything else smelled exactly as it always had. The nose had not dulled. One particular channel had been quietly turned down while the rest stayed wide open. That is a very precise kind of forgetting. Something similar, though partial, happens between related smells. Because a single receptor responds to many molecules, and a single molecule is read by many receptors, saturating yourself with one scent can blunt others that share the same machinery. Perfumers know this state well and call it nose blindness. An hour into a session, the differences between two compositions start to close up, the contrasts flatten, and judgment quietly becomes unreliable. The usual remedy in shops and studios is a bowl of coffee beans to sniff between samples. It is an appealing ritual, and there is no good evidence that it works. When it was tested, people who sniffed coffee beans did no better at picking out a new fragrance than people who sniffed lemon slices or people who sniffed nothing but plain air. The study was small and exploratory, so this is not a demolition. But coffee is not a reset button. It is simply another strong smell arriving in a nose that has had enough of them. And then there is the smell you have carried longest, your own. It is on your skin and in your hair and in the collar of every shirt you own, and other people register it without effort, the way you register theirs. To you it is very nearly nothing, a suggestion at most, and mostly the soap. You can bury your face in your own jumper and get almost no information back. This is the same principle taken to its limit. The one odor that has been constant since before you had memory is the one your brain most thoroughly declines to mention. There is something companionable in that thought. The smells that belong most completely to you are the ones you have handed over to everybody else. They are still there tonight, all around you in the dark, doing the quiet work of making the room feel like yours, noticed by the house, if not by you. Of all your senses, smell keeps the shortest working hours. Hearing stays on duty through the night. A door closing 3 rooms away can still reach you. Touch stays on duty too, which is why a cold foot pulls you halfway up out of sleep. But somewhere in the slow slide from lying still to genuinely sleeping, the nose quietly steps back and for the rest of the night the air can do more or less what it likes around you. This has been tested gently in a sleep laboratory. In one small study, 6 young volunteers slept, while researchers delivered odors through a fine tube at the nostril and waited to see whether anything happened. In the lightest stage of sleep, the sleepers responded most of the time. One stage deeper, peppermint produced nothing at all, and the second compound, a sharp, genuinely irritating one, reached the sleepers on fewer than half of its trials. In the deepest, slow-wave part of the night, neither odor produced a single behavioral response, not the peppermint, and not the second compound, a sharp and genuinely irritating one chosen precisely because it was unpleasant. Through all of it, a simple tone worked. A sound roused the sleepers in at least 3 trials out of 4 in every stage. 6 people is a small number, and the researchers said so themselves. But it points in one clear direction. A sleeping nose is a nose off duty. We wake up and smell the coffee. The coffee does not wake us. There is something restful in that. The eyes have lids. The nose has nothing, no cover, no shutter, nothing you can close, and yet it manages much the same trick by other means somewhere higher up in the part of the brain that decides what is worth reporting. All night the air goes on carrying its usual freight past your sleeping face, and almost none of it is delivered. Almost none. There seems to be one narrow doorway that stays open, and it does not lead to waking. It leads to memory. In 2007, researchers in Germany had volunteers learn the positions of card pairs on a screen, the memory game children play on the carpet, while the smell of roses was in the room. Later, while those same volunteers lay in slow-wave sleep, the rose smell was released again. In the morning, they remembered more of the card positions than sleepers who had not been given the odor a second time, around 97 pairs in 100 against roughly 86. The effect was fussy in a way that tells you something. It worked only if that same rose smell had been present during the learning. It did nothing when given during dreaming sleep, and nothing when given to people who were awake. It helped the memory for facts and places, and left the memory for movements and skills untouched. The picture since then has grown more careful rather than less interesting. When many studies of this kind are pooled, the average benefit is real but small, and smaller still once you allow for the fact that experiments finding nothing are less likely to be published at all. 2 recent attempts using odor cues found no benefit. So the honest position, the one the researchers themselves hold, is that something happens when a familiar smell drifts past a sleeping brain, that it happens in the deep and dreamless part of the night rather than in dreams, and that nobody yet knows how much. Not a technique, just a small open door, still being measured. The dreams themselves are, for most people on most nights, oddly odorless. In one study, 164 people kept dream diaries at home for 2 or 3 weeks and wrote down more than 3,000 dreams between them. Sound turned up in about half of those reports. Smell and taste together turned up in about 1 in 100. Yet when people are simply asked from memory whether they have ever smelled anything in a dream, somewhere between a third and 2/5 say yes. Both figures are probably true. A dream with a smell in it is rare, and rare enough to be kept for years, which is why it feels far commoner than the diaries suggest. Somewhere in your own store of nights there may be one bread, a garden after watering, the inside of a house you have not stood in for 30 years. Meanwhile, the nose is doing something else while you lie there, something you may have noticed without ever quite naming it. The slow swap between your two nostrils has not stopped for the night. It has only taken your weight into account. There appear to be pressure sensors in the skin of the chest and the armpit that take part in it, since resting your weight on one side tends to open the nostril on the other. It is one reason a blocked nose always feels worse at bedtime than it did an hour earlier standing in the kitchen. So the last thing your nose does before it stops reporting to you altogether is turn slowly from one side to the other under the weight of your own body, like someone shifting to get comfortable. Then the reports thin out and stop, and the smells of the room carry on without you, unattended until morning. A dog walking ahead of you on a wet pavement is reading something you cannot see. You take in the hedge, the parked cars, the low gray light. The dog takes in who came this way, and roughly when, and the rhythm of its working, nose down, nose up, nose down again, is the rhythm of somebody turning pages. Some of that difference can be counted. If you line up the olfactory receptor genes of different mammals, a dog carries around 800 intact ones. We carry a little under 400. That is a larger vocabulary, though vocabulary is not the same thing as hearing. What seems to matter more in the dog's case is the shape of the nose itself. Engineers built a detailed three-dimensional model of a dog's nasal cavity and let simulated air flow through it, and what they found is a small piece of plumbing that a designer would be proud of. Air drawn in through the nostrils does not travel as one stream. It splits. Most of it turns downward and goes on to the lungs. A portion peels away upward into a chamber tucked at the back of the nose called the olfactory recess, where the passages narrow into fine scrolls of bone, and the air slows almost to a drift. That slow air is the smelling air. It is held gently over the sensing tissue while the rest of the breath hurries past. Then the dog breathes out, and the design does something else clever. The used air does not leave the way it came. It exits through slits at the sides of the nostrils, angled outward and back, so that the exhaled breath neither washes back over the surface it has just delivered scent to, nor blows away the fragile layer of odor resting on the ground ahead. Sniffing in a dog is close to a one-way current. And because each nostril takes its air from a slightly different patch of the world, the two sides can be compared the way your two ears compare a sound. How sensitive that makes them is harder to say than the popular figures suggest. In one careful study, two dogs, only two, a Rottweiler and a Schnauzer, were found to detect a particular fruity ester at somewhere between 1 and 2 parts per trillion. Different animals are tuned to different parts of the world. The dog was never built to be a better version of you. Farther north on the sea ice, a bear is doing something similar over distances nobody has measured well. Polar bears travel across a surface where there is almost nothing to see, white, then more white, then a low gray sky. What they have instead is a map made of air, drifting up from breathing holes and from open leads of dark water. You will find confident figures quoted for this, so many miles through so many feet of ice, and the honest thing to say is that they are difficult to trace back to any measurement. The bear is remarkable without them. It is crossing a blank page and following a line of scent written across it. In rivers, the same sense keeps time as well as direction. In the middle of the last century, biologists proposed that a young salmon learns the smell of the water it hatched in and carries that memory out to sea. It was tested with a small trick. Young coho salmon were held for several weeks in water dosed with a synthetic chemical they would never meet in nature, then released into Lake Michigan. About a year and a half later, the returning adults could be drawn into an unfamiliar stream simply by scenting it with the chemical they had grown up in. The imprint holds through the years at sea without being refreshed. It is not, as the story is often told, a fish smelling its home river from the open ocean. The open ocean is navigated by other means. It is the last stretch that smell decides, the fork in the water where one channel smells like nothing and the other smells like the beginning. The widest known repertoire of all belongs to the elephant. When researchers counted olfactory receptor genes across 13 placental mammals, the African elephant came in with nearly 2,000 intact ones, more than twice a dog's about 5 times ours. That is breadth rather than keenness. It says how many different molecules a nose might in principle tell apart, not how faint a trace it can catch. Still, it fits what you see. The trunk lifts and turns and hangs in the air like an instrument being tuned. And sharks, whose sense of smell is usually described in terms of a single drop spreading through an ocean, seem to do something quieter and more elegant. The evidence suggests they steer not by which nostril receives the stronger smell, but by which nostril receives it first. A scent arriving a fraction of a second earlier on the left tips the animal to the left. In a sea where currents smear every gradient into nonsense, the order of arrival is the more trustworthy signal. None of this makes your own nose small. It only means the world is being read all the time by a great many readers, each holding the page at a different distance. In a German laboratory across the better part of 20 years, a chemist named Adolf Butenandt worked on a single small question. He wanted to know what substance a female silk moth releases when she is ready to be found. To answer it, his group needed material, and the material was scarce. They obtained the scent glands from something like 500,000 female moths ordered from Japan after the European silk industry collapsed in the mid-1950s. After 2 further years of extraction and separation, what remained at the end of all that labor was 6.4 milligrams of the pure compound, less than a grain of rice. In 1959, he published its identity, and it became the first pheromone ever chemically characterized. He called it bombicol, after the moth, Bombyx mori. The molecule itself is unremarkable to look at on paper. 16 carbon atoms in a chain, an alcohol group at one end, 2 double bonds set at particular places and bent in particular directions. Change one of those bends and the message stops working. That small, specific shape is the entire vocabulary of the exchange. The female makes it. The male is built from the antennae inward to notice nothing else quite so keenly. His antennae are the part everyone remembers. They are not smooth stalks but feathered combs, each branch fringed with fine hairs, so the whole structure behaves less like an organ than like a sieve held out in moving air. Along those hairs sit something on the order of 17,000 receptor cells devoted specifically to bombicol, opening onto an antenna carrying tens of millions of tiny pores. Air passes through the comb rather than around it, and molecules are caught like seeds in a net. The sensitivity of this arrangement is genuinely extraordinary, and it is usually told slightly wrong. A single molecule of bombicol arriving at a single receptor cell is enough to make that cell fire. That much is true, and it is about as close to the physical floor as biology gets. But one nerve impulse is not a decision. The moth's nervous system is already crackling with spontaneous activity, well over 1,000 impulses a second across the antenna, even in perfectly clean air, and a lone signal in that hiss means nothing. In the careful measurements, the moth begins to behave fluttering, walking, turning into the wind, when roughly 3,000 molecules are present in a milliliter of moving air, with only around 6 in every 100 of those receptor cells having fired. So the cell believes one molecule. The animal waits for about 3,000 before it believes anything. Both facts are true at once, and the second one is the more interesting. There is another correction worth making gently, because the picture in most people's heads is of a moth crossing miles of night. The domestic silk moth cannot fly. 5,000 years of being kept and bred left it with a heavy body, weak and flexible wings, and flight muscles too slight and too irregularly arranged to lift it. It flutters and it walks. Its wild ancestor flies perfectly well, and so do the great night-flying moths of hedgerow and forest, and it is in those animals that the long upwind journey actually happens. When it does happen, it does not happen the way we tend to draw it. We think of a scent spreading out from its source in a smooth cone, growing steadily stronger towards the middle, so that all an animal has to do is climb the gradient. Real air does not behave like that. Wind is turbulent, and it tears a plume into threads and ribbons and pockets of nothing, scattered and rejoined. Inside such a plume there is no reliable slope to follow. A moth crossing it meets scent in flickers, some fraction of a second long, separated by empty air. So the strategy is not to follow a gradient, but to respond to the flickers. On contact with a filament, the moth surges straight upwind. Inside the plume it flies upwind with small crosswind zigzags. When the thread is lost, it casts, swinging wide across the wind back and forth until it meets scent again. Its receptor cells can resolve these encounters at rates of up to about 30 a second, and the evidence suggests distance to the source is carried not by how strong the scent is, but by how often the threads arrive. Nearer means more frequent. It is navigation by rhythm rather than by strength. Ants solved the same problem by riding on the ground instead of the air. A forager returning from food lays a chemical line behind her, and other ants follow the line and lay more of their own, so a route that leads somewhere worthwhile grows darker with use, while a route that leads nowhere quietly evaporates. The colony's knowledge lives outside any individual ant, spread thinly across the soil as a smell that keeps or forgets itself. Which leaves the question people always arrive at, and the honest answer is a modest one. We do not have an established human pheromone. Several molecules have been proposed over the decades, mostly steroids found in sweat and skin, and dozens of studies have reported effects, but a careful review of that whole literature found no robust, replicable evidence that any of them functions as a pheromone in us. It may yet be found. For now, the moth has a word, the ant has a sentence, and we have something looser and warmer, a world of smells that mean things to us without ever having been agreed on. A tree that has been cut does not bleed the way you do. In dry country, where the soil is thin and the sun sits heavy on the stones, certain trees answer a wound by pushing out a slow, sticky sap that hardens as the air gets to it. It seals the gap. It keeps out insects and rot, the way a scab does on your own skin. In the low twisted trees of southern Arabia and the Horn of Africa, that sealing sap is frankincense, and from a related tree in the same dry country comes myrrh. Both of them begin their lives as an injury being closed. People learned a very long time ago to make the injury on purpose. A shallow cut in the bark, a few weeks of waiting, and the resin gathers in pale drops that harvesters still call tears. Left in the shade, they turn glassy and brittle, and they hold their smell for years. That last part is the quiet reason a whole trade grew up around them. Almost nothing else fragrant could survive months of travel. A cut flower is finished by nightfall. A hardened tear of resin can cross a desert and arrive still smelling of the tree it came from. So it crossed. Caravan roads ran up the western edge of the Arabian Peninsula and out towards the Mediterranean, and towns grew wealthy simply for being where the water was, on a route carrying something light, dry, and much wanted. In temples, the resin went onto hot coals and turned into smoke, which is where the word incense comes from, the burning. It is a fair guess that the appeal was partly practical, in rooms full of people and lamp oil, and partly the plain fact that the smell is beautiful, warm and slightly bitter, resinous and a little like citrus peel left on a stove. The harder problem was the flowers. A rose gives up its scent to the air within a day of being picked, and you cannot dry it and get it back. The oldest answer was patience and fat, petals steeped in warm oil or animal fat, which slowly takes up the scent and holds it. The more famous answer is distillation. Heat water beneath the petals. Let the steam carry the fragrant molecules up with it, cool the vapor, and collect a liquid with a thin film of oil floating on top, and beneath it the scented water. From that comes attar, the pressed essence of a flower small enough to carry in a stoppered bottle. Tradition gives the refining of that method to Avicenna, the Persian physician and philosopher of 1,000 years ago, working with roses. The attribution is probably too tidy. Distillation is older than he is, and improvements of that kind rarely belong to one person. But the story has stayed with us because it feels right. A doctor bending over a still, trying to keep a garden through the winter. Everything that came afterwards was, in a way, a transport problem. Pepper, cinnamon, cloves, and nutmeg moved along the same logic as the resins, dry, light, durable, and worth carrying, because nothing local smelled remotely like them. Medieval kitchens in the cold north used them heavily, and the taste of the age was for spice on almost everything, which is less about disguising anything and more about the ordinary human appetite for a smell you cannot grow outside your door. By the time you get to the flower fields of grass in the hills above the southern coast of France, the craft has settled into agriculture. Grass had been a town of tanners, and tanned leather smells strongly of the process that made it. Scenting gloves turned into a fashion, and the fashion outlived the gloves. The hillsides went over to jasmine, rose, orange blossom, and tuberose, picked in the cool before the day warms because the scent thins as the sun climbs. Some of those flowers will not survive being boiled. Jasmine and tuberose go flat and cooked if you steam them, and oddly they keep releasing scent for hours after picking, as though nobody had told them. So the growers used the fat. Purified lard and tallow were spread smooth over sheets of glass in wooden frames, fresh blossoms laid across the surface by hand, the frame stacked and the flowers changed the next day, and again the next for weeks until the fat was saturated. Then the fat was washed with alcohol, and the scent moved across into the liquid. It was called enfleurage, and it was slow, expensive, and almost entirely done by hand. Then chemistry arrived, and the shelf got bigger. Coumarin, the sweet smell of new-mown hay and dried woodruff and tonka bean, was made in a laboratory in the 1860s, one of the first fragrance molecules built rather than extracted. In 1882, a perfume appeared that used it openly, and it was called Fougère Royale, Royal Fern. Ferns have essentially no smell at all. The perfume was a portrait of something that had never existed, hay and moss and a cool green shade, an imagined forest floor. That is what the synthetics really gave. Not cheapness, though they gave that too, but access to smells that no flower will hand over, the note that cannot be distilled, the plant that yields nothing, the thing you only ever smelled once. A perfumer could now work with the memory of a smell rather than only with the plant. There is a smell in the room with you now. It is probably a quiet one. Rooms rarely announce themselves, and yours has long since stopped announcing itself to you because you have lived inside it, and your brain has decided, quite sensibly, that nothing here needs reporting. But it is there. The pillow under your cheek has a smell. Warm cotton, a little of your own skin, the faint sweetness of laundry soap that stopped being noticeable within an hour of the sheets going on the bed. If you turn your head slightly, the pillow gives a little of it up. That is often all it takes. A small change, a fresh patch of fabric, and for a second or two, the scent comes back before it settles again into the background where it belongs. There is the air of the house beyond the door. Wood and dust and whatever was cooked here today, still hanging faintly in the hallway. There is the cold thread of outside air coming in around the window frame, thinner than indoor air because molecules leave surfaces slowly when it is cold, and the night holds less scent than the afternoon did. Winter air smells clean partly because winter air is empty. And if it is raining tonight, somewhere under all of that, there is the smell of wet ground. You know now what that is made of. An oil that has been quietly gathering on stone and clay through every dry day, waiting. A bacterium in the soil that makes a molecule so distinctive that your nose can find it in a few parts per trillion of water, and yours may find it more easily or less easily than anyone else's. A raindrop landing, and in the moment it lands, throwing a fine mist of the ground back up into the air. And the gentlest rain is the most fragrant. Heavy rain hammering down makes far less of that mist than a soft, steady fall does. So the rain that is easiest to sleep through is also the rain that smells the most. That has been true on every quiet night of your life, and you did not need to know it for it to be true. There is bread somewhere in your memory too. Perhaps a kitchen, perhaps a bakery on a street you have not walked down in years. The crust of a loaf makes a molecule in the heat of the oven, and that molecule is not a cousin of the smell of jasmine rice steaming under a lifted lid. It is the same molecule. Bread and rice and popcorn and pandan leaves all reach for you with the same small chemical hand. There's a kind of tidiness in that which is pleasant to think about while falling asleep. Wood smoke is out there somewhere in the world tonight, and the compound doing most of the work in it smells, on its own, of cloves and vanilla. The chemistry of a fire in winter really does lean towards comfort. That is a coincidence, of course. But it is a good one. Here is a last small thing, and then you can let go of all of it. The cells at the very top of your nose are new. They are replaced through your whole life, a rolling renewal, so the surface that meets the air tonight is not the surface that met it last spring. But the structure just behind them, the pair of small bulbs sitting under the front of your brain, appears to be original. The evidence we have suggests those neurons are as old as you are. They were there for the first rain you ever smelled. They are here for this one. So the part of you that greets a new smell keeps being made again. And the part of you that keeps them has been yours since the beginning. You do not need to hold on to any of this. The nose is the one sense that clocks off at night, more or less entirely. Sound will still reach you. Smell will not. Whatever the room does while you sleep, it will do without telling you, which means you can stop noticing now. Let the pillow be warm. Let the soap be faint. Let the rain, if there is rain, do its work on the stones outside without you. Somewhere a bakery is cold and dark and will be lit again in a few hours. Somewhere a fire has gone down to embers. Somewhere the ground is holding its oil and waiting for weather. None of it needs you tonight. Your breathing has probably already slowed. In and out, through the two nostrils that have been taking turns all evening without your knowledge, quietly dividing the night's air between them. The room smells of itself. It always has. You are warm. You are still. Nothing here requires your attention. Let your shoulders go. Let your jaw go. Let the last thought unfinish itself. The rain will keep falling, or it will not. The morning will smell of coffee, or of cold air through an opened window, or of nothing much at all. That is tomorrow's business. Tonight there is only this: a pillow, a trace of soap, a little cold air at the window, and a mind that has stopped needing to name things. Rest now. Good night.